Thank you so much, Professor Jean-Marc Burgunder, for joining us today. We are very excited to hear about the work that you're doing within the field of gene therapies, and in particular for rare neurologic diseases. Gene therapies as a whole has evolved a lot in the past 10 to 15 years, going from a topic that many people were reluctant to touch to being a topic that excites many millions. So with that, I'd like to hand over to you and hear more about what you can tell us.
Well, you're right, this is very exciting indeed. And as a matter of fact, it starts with a definition. What is actually gene therapy? And gene therapy might be thought as going to repair the gene, actually. But I would like to take a slightly broader perspective in Yes, now it's working even better. So to take a broader perspective at the definition of what is gene therapy, instead of thinking only of gene repair, which is possible now indeed in some rare occasions. It's possible in animal models, but there are now some diseases, maybe not precisely brain diseases, but several diseases from the eye where machinery can be constructed with molecules and put into the eye, which will go and cut a piece of a gene which causes blindness in people and repair this, and people will be able to see again. So this is possible in such an occasion because it's very easy, or slightly easier, or much more easier than putting some of this construct of this machinery to repair a gene into the eye than it would be into the brain, you can imagine. So this would be the strict sense of a gene therapy. However, gene therapy in a broader sense can be thought of intervention in all the parts from the gene itself, which is mutated, and this is frequently the case in rare diseases. These are genetic diseases, very often, not always, and not only to think about repairing the gene, but intervening on what occurs after the information of the gene is being, first of all, transcribed in what we call the messenger RNA, which is, again, a substance which is now present in the cells and which can be directly accessed in using substances which will recognize this piece of information, which is a molecule, and this piece of information will be recognized by what we call antisense oligonucleotide, and this one would hinder that information to be passed further in translation, which is constructing the actual protein for which the gene has information. So you have the gene, you have this piece of information, and then you have the protein. Now, as I said, repairing the gene starts to be possible, is possible also in animal models. The difficulty here is that we need to be more precise in the targets, both in targeting into the brain at the right place, as well as targeting the gene which wants to repair, instead of then addressing other genes which will then be disturbed. So the second step which can be addressed is the one using antisense oligonucleotide, which will recognize this information, this transcript of the gene in the cell, and which then would hinder the information to construct the protein. And these have been the field of a large number of studies now, particularly in Huntington's disease. This is my major field of interest, is Huntington's disease, where this has been done. And there were quite a large excitement in the field to be able to now do this after having animal studies and then preliminary studies in human, where indeed the amount of that protein could be decreased in using this method. Unfortunately, in a large study which encountered 800 people, which was run very quickly, it was an excellently run study, unfortunately, the effect of having a change in the course of the disease could not be achieved. But there are other diseases where it has been, and other studies are still going on with this type of intervention. Finally, the intervention could also be at the level of the protein itself. So you have a gene which is default as a message which is as a mistake, and then you have a protein which has a mistake, which will lead that protein to be increased at the place where it deposits, or which will be unfunctional. So you could, if it is unfunctional, a lack of function, like for example enzyme, what is possible is to use a construct to prepare a protein outside of the body, for example, with cell of the person or in another system where this is being developed, and then put this enzyme into the body at the appropriate place where it would replace the missing function. And gene therapy can even involve also small molecules which would also interfere. It is a flow of information from the gene to the protein. And this is what I will speak about during my talk later during the Congress.
Thank you so much. That was a really good overview of the different steps and processes that are possible. If we take it a little bit back from that scientific level to the human level, and we think about clinical trials, and step one, I would imagine, is actually obtaining a genetic test or an accurate genetic diagnosis. Is there any difficulty with recruitment when it comes to genetic testing for certain conditions? Is there a reluctance? Because sometimes this is one side of the coin that we hear. The other side is that there are many people who would love to be part of clinical trials, but they don't know how to be involved in a clinical trial. And is it very dependent on the relationship that people have with their general practitioner and then their specialist, their neurologist? Could you speak to us a little bit about that process?
Oh yeah, for sure. And I can provide you with two examples which I've been experienced myself in a recent couple of months. You're right, it starts with gene diagnosis. So of course, if someone wants to participate in such a study, needs to have a gene diagnosis. So in the context of Huntington disease, the gene is known for a long time already, and the people start to want to have a genetic testing even before they have the manifestation of the disease. Now, of course, if there is no treatment at this point of time, not a lot of people want to actually take this opportunity to have the genetic test being done. But the reason why the studies in Huntington disease have been so successful in terms of participation is that we had prepared for years, for decades, a cohort of potential participants in such a study. The idea was not so much to prepare the further study on treatment, the clinical trial, but the reason were, first of all, to have a better understanding of the phenotype, the presentation of the disease. So globally in Europe and also in North and partly in another country in South America and the world, we have developed large cohorts, and now we have merged these efforts to have one single worldwide cohort of patient and person at risk to having Huntington disease. We have now 22,000 participants, active participants in such a study. So it's a tremendous number. And so at each center, you would know a number of people, and it was possible to recruit for this Roche study, the one I just mentioned, which unfortunately didn't show up exactly what we wanted. It was possible within weeks to recruit so many patients and run the study. The other example I want to do, which is in my place, unfortunately, not well organized, this is in spinocerebellar ataxia. So a pharmaceutical company, knowing that we were involved in this type of studies for Huntington's disease, and you may know that there are some similarities in terms of the genetic changes, which is another gene, but a similar phenomenology, which occurs at the level of the gene. So they asked us, we would like to run a trial, a similar trial with an antisense oligonucleotide. You remember this one, which will block the transmission of the information to the protein. And so they asked whether we would be interested in participating in a trial on SCA. And I said, of course, I would be interested. So, but at this point of time, I didn't have so many patients coming to my clinics, because I had a research project before, but it didn't continue. So I tried to find out in Switzerland, in my country. I asked all people whom I know, because I know all of them, who would be treating this patient. It was not possible to have like even 4 or 5 patients in running. So as you say, there is, of course, an interest of the participant, but there must be also structure where they can actually get in, and their involvement and their interest and enthusiasm to participate in the studies is great.
Can I ask a little bit more about that? When it comes to the design and the development of clinical trials, do you think at present there is a role for patient involvement in the design, and what level is that at present? And I'm going to ask that with an add-on. When it comes to information we've generated this year at EFNA, covering a broad spectrum of neurological disorders, when it comes to receiving a diagnosis and going through any sort of therapeutic intervention process or care pathway, the majority of people, over 80%, indicated that they would have had a preference for having some sort of counselling to accompany that diagnosis and treatment and care pathway. I can only imagine that this is even more important when it comes to potentially inherited conditions and genetic counselling. And is that incorporated throughout the clinical trials that you're aware of?
Information and care is the most important part of it. And a clinical trial should, from my perspective, be embedded completely, as much as possible, within this care setting. So it might not be that each part where people get this care, this comprehensive care with the care pathway, that they would all participate in clinical trial, of course, because they are maybe not, it's not possible, but that this would still be embedded there. Or put it in the other way around, even starting before genetic counseling or at the time of genetic counseling, one should always have in mind this care pathway, like in Huntington's disease, having in mind the family. So maybe I have seen already the parents, the grandparents, or the counselor who comes now, but I know it may take time until the disease actually comes, but still the expectation is there, and the concern of the people is, now I have the disease because of that symptom. So it's very important to accompany them, and then the study comes like naturally in it. For example, this Enroll-HD study, which is the observational study I mentioned, is with us embedded in our clinical care. So we use the same instrument to examine how they are doing now and provide them with the treatment. And we use this same data, of course, with informed consent to use in the database. And when it comes to a clinical trial, this database will help to find out the patient and will help me to ask this one patient, look, you might be interested in that. And they have already provided consent that I would ask them if something comes like this, that I would answer. So yes, patient and community involvement, and also and family involvement is of utmost importance, not only in this context I just mentioned, but also in the preparation of the protocol itself. It's important to understand what are the needs, and if we measure something which eventually is not something which is disturbing the patients, maybe is not so important. We need to find out what is important in terms of clinical trial in general, not only disease-modifying ones, but also symptomatic treatment. What are the symptoms, and what are the problems which people are and their family are expecting, and not what we feel as scientists is what is obvious.
If I may go back to the start of your discussion, communicating what gene therapies are and what they can do to the human body, do you find that that is something that is easily understood by the patient community, or do you think as patient organisations we still have a bit of work to do in ensuring that that's translated as efficiently as possible?
We, and you said the patient organization, but I think we together with the patient organization, we I mean the so-called specialists, should definitely a joint effort to explain the disease, to explain what is involved in participating in a clinical trial, and to be clear, a consultation with a family or with a patient with Huntington's disease takes maybe kind of a couple of minutes of explaining, the patient explaining to me what is the problem, and more minutes, and then it takes a very long time to actually answer those questions and explain what is all about in trial, and even also to the families. And this is worthwhile to take it because one needs to find out the right language to explain, and this may be at several levels and definitely needs to be done and redone at many occasions. It's very important, and this is a joint effort which community and us actually being part of that same community have the responsibility to do.
I see some very happy, smiling faces when you have said that, so that's music to our ears. If I might ask you one last question, and maybe you're saving some special insights for your talk later today, do we as a community, as the neurology community, and indeed those who are involved in the rare neurological disease space, do we have a reason to be hopeful for what's coming down the pipeline? From your insight, do you think that we will see more and more gene therapies being approved? Is this something that we can have a hope for?
My answer is certainly yes, but a yes which needs to be qualified. We were very, very excited with this study on Huntington's disease, and we thought, well, it is clear. We have something now at hand. We were even already going, and myself, I was excited and did that work and presented that work. What shall we do if this study is successful? Are we going to have enough centers who will be able to apply that medication? Because it is a lumbar puncture and it's not very simple. So we made studies out of this, what is the burden for the centers, and so on and so forth. As excited we were. And of course, then it came that the study was not, didn't show up the endpoint we wanted, and the question then was how to explain that. So, but the answer is still a yes, because even on that, the company which has run that study is continuing to be involved in Huntington's disease, even preparing a new study with the same compound with another group of people. And there are other companies which also continue, despite also their drawbacks, and this is only part of it. And the other thing which I didn't say before, but I will go more into my talk, is that there are now, when you look at the clinical trial org, this is the place of the US government where all the studies are being listed, there is now about 15 different studies using vectors to prepare like these enzymes for replacing what the enzyme which is disturbed in the body. So now on the pipeline in this and last year. And there have been 2 new approvals for such therapies at the EMA, one which is an approval and the other one which is suggested to be approved. So this is the latest, last week. So this is encouraging. Now, of course, we need to keep rational, and we would like, and the community would like to have answers very quickly. And we have been waiting now 25 years since knowing the Huntington's gene, and only now we can do this study. So my answer is certainly yes. But let's be patient and let's do what else we should do in taking care and in communicating and helping the community with what we now have at hand.
Thank you. And And with that word of excitement, tipped with a word of caution, Professor Jean-Marc Burgunder, I'd like to thank you so much for your time and your participation today in our event. And we do look forward to hopefully some very exciting and helpful therapeutics to come.